Welcome to Circuit Buddy. Today we will be discussing about glycolysis. Glycolysis is a very important topic of class 11 biology. It's also important for Neet. So let us discuss about glycolysis in details. Keep watching the video till the end so that you can understand and memorize all the important topics of glycolysis. Glyco means carbohydrate and lysis means breaking. So it is basically the process of breaking down carbohydrate molecules deriving ATP and NADH+. Okay, so it is a catabolic reaction. Here you can see that in the first step, glucose molecule is converted to glucose 6-phosphate by the action of the enzyme hexokinase, and here kinase means ATP will be hydrolyzed or generated. So ATP, one molecule of ATP is hydrolyzed to ADP and the inorganic phosphate is attached to the 6th carbon. Okay, so that is glucose 6-phosphate. After that, glucose 6-phosphate is converted to fructose 6-phosphate by the action of the enzyme phosphohexose isomerase. Okay, so isomerase is means enzyme, and as it is an isomer, so it is isomerase. So fructose 6-phosphate is then converted to fructose 1,6-bisphosphate. As the name implies, you can easily understand that in the 1 or the first or the 6th, in the 6th position, phosphate groups has been attached, okay, by the action of the enzyme phosphofructokinase 1. As the name implies, kinase means an enzyme which hydrolyzes ATP or generates ATP. Okay, so it is actually basically transfers phosphate groups. Now the ATP is hydrolyzed to ADP and the phosphate group is attached to the first carbon, okay, forming fructose 1,6-bisphosphate. After these, fructose 1,6-bisphosphate is converted to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Okay, so it is clipped in two parts. One is glyceraldehyde 3-phosphate and the other is dihydroxyacetone phosphate by the action of the enzyme aldolase. Okay, so if you see the open chain structure of fructose 1,6-bisphosphate, here you will see that this portion is clipped, okay, and here an aldehyde group is introduced, and here you can see that this carbon takes another H, forming CH2OH, and this is the structure of dihydroxyacetone phosphate. Okay, so dihydroxyacetone phosphate here one which is there. Okay, now it is the preparatory phase of glycolysis. Now we will be talking about the payoff phase of glycolysis, in which we will see that the glyceraldehyde 3-phosphate and dihydroxyacetone phosphates are utilized to form ATP and NADH+. Okay, so in the first step, what happens? Dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate by the action of the enzyme triose phosphate isomerase. So let us see. So here you can see that dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are there, and the dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate by the enzyme triose phosphate isomerase, forming two molecules of glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate. Now glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate by the action of the enzyme glyceraldehyde 3-phosphate dehydrogenase. Okay, so first of all it is converted to 1,3-bisphosphoglycerate. Okay, so phosphate OH, again you have CH2OP. This is 1,3-bisphospho, so two phosphate groups are there, phosphoglycerate. After this, 1,3-bisphosphoglycerate is then converted to 3-phosphoglycerate sorry, the enzyme name is glyceraldehyde 3-phosphate dehydrogenase. One more important point is that whenever there is a dehydrogenase, it means it is removing the hydrogens from the substrate, okay, and here the hydrogens are accepted by NAD plus, forming NADH plus, okay, and the extra phosphate that is coming here in the molecule, it is actually inorganic phosphate, okay. Now 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate, so it is converted to 3-phosphoglycerate. Phosphoglycerate. Let me draw the structure. Okay, this is 3-phosphoglycerate. Now this reaction is catalyzed by phosphoglycerate kinase, and as it is a kinase, it will form ATP here, because one phosphate is removed, ADP is converted to ATP. After that, 3-phosphoglycerate is converted to 2-phosphoglycerate. So 2-phosphoglycerate, that means the phosphate group is coming to the second position, okay, and the third position we have, okay. So that is basically 2-phosphoglycerate, 2-phosphoglycerate, and the enzyme that is catalyzing the reaction is called phosphoglycerate mutase, okay. After that, 2-phosphoglycerate is converted to phosphoenol pyruvate, okay. So let us draw the structure. So phosphoenol pyruvate, there is the enol enol group introduced here. So this is basically phosphoenol pyruvate. We write it here, phosphoenol pyruvate, okay, and the enzyme that is catalyzing the reaction is enolase, okay. One more important point that I missed is that when we are converting 2-phosphoglycerate to enolase, to phosphoenol pyruvate, the phosphates are in the same position, no change in the location of the phosphate groups. So these points should be remembered to draw the structures in a correct way, and after that phosphoenol pyruvate is converted to pyruvate, and it is the last step of glycolysis, okay. So pyruvate, double bond O, that means the phosphate is removed, and this is CH3, okay. So this is pyruvate, and you can see that this reaction is an irreversible one, and it is catalyzed by pyruvate kinase, and as it is a kinase, it will produce ATP from ADP, because the phosphate has been removed from the molecule. And now this pyruvate will enter the Krebs cycle, okay. I have made a separate video on Krebs cycle, do watch the video. If you like the video, please like, share and comment. And thank you very much for being with me to the last of the video. Thank you very much once again. Have a nice day.